Check, check. Hello? Can anybody hear my voice? Check.
Yes.
Okay, thank you. Good evening, all participants. We will soon start our opening ceremony. No, I'm sorry. We will soon start our internal conference in day 2. But before we start to plenary session 2, please be reminded that you have a field attendance form for BaSiC in any session that we have informed before. And now we shall commence this plenary session. Assalamualaikum warahmatullahi wabarakatuh. Shalom. Om Swastiastu, Namo Buddhaya Rahayu, may peace be upon us. The honorable speakers, Professor Andrew Clayton from Swinburne University, Professor Terry Mark from Universitas Indonesia, and also Associate Professor Dr. Aji Ahmad Rinaldo Fernandez from Brawijaya University. Distinguished participants, and also ladies and gentlemen, good morning and welcome back to our event, the BaSiC Science International Conference 2022 on day 2. Today we will have the plenary session 3 and 4, as well as online parallel presentation with each link we have shared before. And at the end of this conference, there will be closing ceremony and also awarding of the best presenter. Well, honorables, distinguished guests, ladies and gentlemen, let us begin our plenary session 3 with our speaker, Professor Andrew Clayton from Swinburne University, Australia, and Professor Terry Mark from Universitas Indonesia. This plenary session 3 will be led by Ibu Zubaidah Ningsih, PhD. Time and screen are yours now.
Thank you very much, Ibu Jilda, for the introduction and the opening of the second day of the BaSiC conference. And for this plenary session, we will have 2 speakers. First is Professor Andrew Clayton. Hello, Andrew.
Hi, Zubaidah. How are you?
Fine, thank you. Nice to meet you again, Andrew.
Nice
and
to meet
also,
you
yes, the second is Professor Terry Mott from Universitas Indonesia. I think Professor Terry also already in Zoom. Thank you very much.
Yeah.
And for the first session, we will have Professor Andrew Clayton from Swinburne University to present his talk this morning. Not yet. Okay, so let me introduce Professor Andrew Clayton. So Professor Andrew Clayton is my former supervisor. So I will introduce him as a very supportive, humble, and knowledgeable supervisor. And he's also in Optical Sciences Center, Swinburne University of Technology. He's graduated from Melbourne University, Australia, and he's one of a very reputable author in biophysics. Okay, shall we start, Andrew?
Sure.
Okay, thank you. Time is yours.
Thank you so much, Zubaidah, for the invitation to speak and for the very kind introduction. What I'm going to talk about today is detecting intermediate states in ligand-protein interactions using edge excitation spectroscopy. So I hope to debunk what that title means throughout this talk. So just acknowledge the people that were involved in this research, PhD student Lutful Kabir, PhD student Mohammed Khattab, and also a colleague from the Department of Chemistry and Biotechnology, Professor Fen Wang. The scale of things I should mention here, our labs are cell biophysics labs, so we're interested in objects, cells of the order of 10 microns in diameter, But in particular today, I want to talk about proteins, and proteins are of the scale of about 1 to 10 nanometers. So let's just give an idea about size. And when we talk about cells, cells really represent the smallest unit of life, of course, and the functioning of cells is linked to health and disease. So if we think about important diseases such as cancer, antibiotic resistance, and Alzheimer's disease, these can all be understood in terms of what goes wrong with cells. In cancer, for instance, we have uncontrolled cell growth. In antibiotic resistance, you have the appearance of new strains of microbes that are not affected by current antibiotics, and that's a very big problem in infection. And then in Alzheimer's disease, you've got causative agents in the brain that attack neuronal cells, causing memory loss. Now, the thing that's common to all of these things is if we look inside our cells, so there's about 100 trillion cells in the body, and within every cell of our body, we have all of these proteins and DNA, membranes and nucleic acids and carbohydrates. And what's common is that we have these interactions between these molecules. And these interactions, these non-covalent interactions, largely dictate the functioning of these cells. And a lot of the drugs that we take to help a lot of these illnesses actually block these interactions. So let's look at protein ligand interactions perhaps in more detail. So if we represent, say, a protein here by, this looks like a Pac-Man, and then a ligand by the piece of cheese here, these can be free in solution. But also, if there's a net free energy gain from them coming together, they can form a complex. Here you can see the piece of cheese is now in the mouth of the Pac-Man. And from a very simple kind of model is that we have concentration of protein plus a concentration of ligand, and then that forms the protein ligand complex. The efficiency of this process is related to the relative on rate and the relative off rate. So the so-called equilibrium constant is K on divided by K off. And at equilibrium, of course, the affinity is also defined by the concentration of the complex divided by the product of the concentration of the three parts. And you can see here that if we had a solution where everything's in equilibrium and the majority of what we had in solution was the complex and we didn't have much of the free components, then obviously K is a very big number. That's a high affinity interaction. On the other hand, if we had a low affinity interaction, the majority of what we would see in solution is just the free parts here. And biochemists often use not just the affinity constant, but they talk about the equilibrium dissociation constant. This is just the inverse of K, and it has units of concentration. So high affinity would be something like picomolar or nanomolar. Low affinity would be millimolar or molar. Now, affinity is important because it tells how strong an interaction is between two things. And also, it's important in drug design because we want to be able to design a high affinity drug that binds into the protein site that is then more effective. But if we look at how protein ligand interactions occur, there's really three kind of models. There's a lock and key model where we have one state of the protein and one state of the ligand, and they form a reversible association in solution. So the ligand here sort of locks in to the kind of key, if you like, or the key locks into the lock, and you have a very stable complex being formed there. That's the lock and key model, which is of Emil Fischer, which is a very old model. But that model is often in textbooks, but it's not quite the whole situation because we know proteins can exist in different conformational states. So in the conformational selection model, you have a protein that exists in two different conformational states. One state it doesn't bind to the ligand, the other state it does. And there's an equilibrium between those two states. There's an interconversion between the two conformations in solution. And when the ligand comes along, it just binds to the ligand competent conformation that forms the ligand protein complex. And what you can see is that there are two free protein states and only one state of the ligand protein complex. In the induced fit model, you still have different conformational states of the protein, but the way they're achieved is slightly different. In this case, if you like, the ligand binding actually induces a conformational change in the protein. So in this case, there's one conformation that's free, and then there's two states of the ligand protein complex, one where the ligand is partially bound, and another state where the ligand is fully bound. Now, the importance of an intermediate state in the context of this induced fit model is it provides a rationale for understanding not only the affinity of the interaction, but also the lifetime of the protein ligand complex. And so affinity is very, very important, but what's being recognized by people who do a lot of pharmacological studies is that the lifetime of the complex is also a very important determinant. A longer lived complex is going to be more effective than a short-lived complex. A long-lived complex is going to be a better way of separating, let's say, off-target interactions than a short-lived complex. And one of the things that determines the lifetime of the complex are these intermediates. So if we consider the model here, I have a protein and a ligand, they're forming a complex, and there's an intermediate state where the ligand is partially bound. Now, this is higher energy, so it's not populated very much and it's only transient. But once you get to that state, the system has a choice either to release the ligand and form the protein like this, and the ligand's free in solution somewhere, or it can fall back down into the more favorable energy state, which is the bound state. So you can see that having an intermediate there acts like a little bit of a kinetic trap. It sort of keeps the ligand associated with the complex for much longer. Now the question is, so now the study is not just, or now the field has switched, so we're not just talking about studying the final state, but we're also interested in detecting the intermediates. So how do we do that? This is very challenging. The reason is intermediates have a low population at steady state, and intermediates are transient based on what I just said before. Because the intermediate is high in free energy than the final state, bound state, then it's going to be less populated. The other thing is we form it and then it rapidly goes back to the initial state, or the ligand is released into solution. So it's only transient. So that it's hard to detect these. So the techniques that are available are NMR. I won't go into too much detail about that. Single molecule fluorescence is another way of detecting it. So you have a laser beam shining on a dilute solution of molecules, and as the molecules pass through the laser beam, you get a blink of fluorescence. Maybe you can detect the intermediates that way. There's stopped flow kinetics, where you have the ligand and the protein, you flow them together, and then you measure the kinetics of the interaction. From that kinetics, you can then maybe detect one or two intermediates if you're lucky. And then what we're going to propose today is to use just simple optical spectroscopy, absorption and emission spectroscopy, and this technique called REES, red edge excitation shift, to see intermediates. So why do we use fluorescence? Just to remind you that in fluorescence, the probes are very site specific, the signal's sensitive to environment, there's multi-dimensional information content, and fluorescence is very sensitive to changes in structure and dynamics. The multi-dimensionality is illustrated in this busy picture here. I won't go into all of the detail, but what I want to focus your attention on is the emission spectrum. This is an example here of an emission spectrum of amino acid tryptophan. When the protein is in its native state, we have an emission spectrum here, but then we get a large shift in the emission spectrum when the protein becomes unfolded. And so the fluorescence is really, really sensitive to the environment that the fluid was in. So the main thing I want you to remember from this is that the fluorescence spectrum is sensitive to environment, and we're going to use this to detect intermediates. Now, what is red edge excitation shift? It stands, REES stands for red edge excitation shift. It was discovered independently by 3 laboratories in 1970. So it was actually Gregorio Weber and Rubinov and Gallet. So Argentinian, a Canadian, and I think someone from the Soviet Union would have been in 1970. And what they observe is if I change my absorption wavelength, so I go to the peak of my absorption spectrum, and then if I shift the absorption wavelengths to a slightly longer wavelength, what I see is my emission spectrum also shifts, right? My emission spectrum also shifts. And this is a really interesting phenomenon. It results from having a distribution of environments around the chromophore, around the fluorophore, but also what's important is that the environment around the chromophore is very rigid on the fluorescence timescale. So the only way that you can really observe this is if you have some rigidity of the environment. Now, I'm just going to give you an example of how you might, how this phenomenon might arise. So the most important thing is that the fluorophore has to have a difference in dipole moment between the ground state and the excited state, right? So that means that the fluorophore interacts with the environment differently in the ground state and the excited state. You need some sort of heterogeneous environment. And the third thing is that the environment around, the molecules around the fluorophore can't relax on the fluorescence timescale. These are the 3 requirements. I'll just give you an example of a sort of simple toy model to illustrate what I mean. So let's suppose I have a fluorophore and I represent it by the symbol DA, and I've put, which really means donor and acceptor, and I've put a positive charge on the donor and a negative charge on the acceptor. This is just to indicate that there's a finite dipole moment of the fluorophore in the ground state. Note that when I go from the ground state to the excited state, the charge distribution changes. So here there's a plus and minus, and in the excited state, the charge distribution changes. So this is a requirement of this. There has to be a change in the charge distribution going from the ground state to the excited state. Now, suppose I put a charge, a positive charge, just to represent one of the environments close to the negatively charged A here. So because this is a Coulomb, this is an attractive thing to do, plus and a minus go together, then the ground state is going to be very, very stable. This is going to stabilize the ground state. It won't do much to the excited state because the excited state is neutral. So the excited state stays the same, the ground state is stabilized. And so when I have an absorption photon here, this is going to be high energy and short wavelength, and the emission is going to be high energy, short wavelength. Now suppose the fluorophore is in a slightly different environment, now with a positively charged close to the D+. Now a plus close to another plus is kind of repulsive in a Coulomb sense. And so now the ground state is shifted to higher energy. The excited state won't change very much because this is neutral, so that stays in the same position. So the net result is now there's a small energy gap between the absorption, the ground state, and the excited state. So this photon will be absorbed at smaller energy or longer wavelength, and the emission also will be at longer wavelength as well. So we can see that if you have a distribution of environments, we're going to have the most populated state, which will be the peak wavelength, is going to be at a short wavelength, so short wavelength here, whereas the less populated state is, which will be, say, somewhere here, that's going to be at a longer wavelength, right? So I'll get short wavelength and I'll get a blue emission, long wavelength, which is less populated, I'm going to get a red emission. So what I'm proposing is that we analyze this REES experiment. So the idea is we have a solution method. We can do this for proteins in solution. We need simple instrumentation, just a UV vis absorption spectrometer or fluorescence spectrometer. We get quantitative data because we're measuring the emission as a function of the excitation. We're plotting the peak emission wavelength against the excitation wavelength. And then we fit the data to a theoretical model. So we have a free energy difference between a bound state and an encounter state. We get the number of intermediate states. We also get the nature of the free energy landscape, whether it's linear, quadratic, or cubic. Now, the theory, I'm not going to go into detail into the theory, but this is described in this paper here that we just published last year. So the particular protein targets that we're talking about are these things called tyrosine kinases. And tyrosine kinases are enzymes in every cell in our body, and they're linked, some of them are linked to cancer. So they're linked to uncontrolled cell proliferation. They're so important that the US Food and Drug Administration approved 49 so-called tyrosine kinase inhibitors as anti-cancer drugs in 2019. So this is one of these anti-cancer drugs, this tyrosine kinase inhibitors. It's called AG1478. And this is a preclinical drug, but some of the drugs that are actually used in the clinic are based on this structure here. They have a quinazoline ring and they have an aniline ring here. And it turns out that these drugs are fluorescent. They're not fluorescent in water, but they are fluorescent when you attach them to a protein or put them in a non-polar solvent. This is a plot of the fluorescence spectrum of this drug as a function of different solvents. So in the red spectrum here, we have an emission maximum of about 400 nanometers. This is a spectrum in a non-polar solvent such as toluene. And then if I measure it in a very polar solvent such as DMSO or dimethyl sulfoxide, you can see that the spectrum shifts from 400 nanometers all the way to 475 nanometers. So that is a huge shift with polarity, very polarity sensitive drug. And that makes it very, very important for when we want to detect intermediates. Here's some spectra of this drug when it's bound to two different proteins. So both of these proteins are enzymes. They both have the fold of a eukaryotic protein kinase. So this is called mutagen activated protein kinase 14. So this is important protein in pancreatic cancer. This is an aminophosphotransferase. This is from bacteria. This turns out to be an important protein for antibiotic resistance. But they both have the same fold. So they have both have the same three-dimensional fold. But strikingly, if you put these anti-cancer drugs in the ATP binding site here, they have completely different spectra. You can see here, this is the spectrum in the MAPK14. It has emission maximum at about 420 nanometers or so. And then if we look at it in the aminophosphotransferase, it has a completely different spectrum. Here it's at about 475 nanometers. So they're totally different. The environment's totally different. The other thing that's interesting, perhaps, or to note, is that if we look at the spectrum as a function of temperature over a range of 15 to 37 degrees here and 10 to 37 degrees there, the spectra don't change very much. What that tells me, or the emission maximum doesn't change very much, what that really means is that the protein drug complex has remained intact through those different temperature changes. And you'd expect that. These things are supposed to be functional at 37 degrees, right? So we don't expect them to unfold in those temperatures. Now these are the so-called red edge excitation shift spectra that we got, and this is published in Journal of Physical Chemistry in 2018. So what we're plotting is the fluorescence maximum. So this is the peak of the fluorescence spectrum as a function of the excitation wavelength. So if I excite at 330 nanometers, which is kind of the peak of the absorption, I get an emission at around 423 nanometers. If I then excite at the very red edge of the absorption here, I get a peak maximum now at 436 nanometers. So there's a whopping shift of about more than 10 nanometers in the emission when I excite at the main peak or at the red edge, right? So this is a really remarkable thing. I wouldn't expect to see this if I had this fluorophore in solution. If I put this in solution, I don't see that. I see that the emission spectrum is independent of the excitation. In this case, it does depend on the excitation. And what that means is that this is an emotionally restricted environment or set of environments inside the protein. The protein, the dipoles in the protein and the atoms in the protein are not moving very much on the nanosecond timescale, which is the timescale of the fluorescence. If I then look at this protein here, which is the immunophosphotransferase, you can see also it exhibits this Reese effect, red edge excitation shift. Starting and the final wavelengths are different, but there's a very, very big shift here of nearly 30 nanometers. That's a huge shift in the spectrum when we go from the peak to the red edge. The other thing to note is it's not very temperature sensitive, right? And that means because it's not very temperature sensitive, between 10 degrees and 37 degrees, what that tells us is that what's causing that red edge excitation shift is due to heterogeneity in environments, not due to any dynamics that's occurring, heterogeneity. So this is our picture of what we think is happening. So this is a kind of free energy landscape idea of what we think is happening, that the ligand is in different states of the protein. So it's in the most bound state or in a sort of very sort of semi-bound state. And those two different states have different environments. So this is why we have the red edge excitation shift. But this state is higher in energy than this one, right? And this means this is less favorable. And if it's less favorable, then it's on the red edge of the absorption spectrum effectively, because this is the most favorable state. It's the lowest energy state. This is the one that we're going to be exciting when we excite the main part of the absorption. So this now is what we're plotting here. It just fits to various theoretical models for this kind of free energy landscape that I just showed you. So the data points are the experimental points and then the lines for different models. What we can also get out from this is the number of what we call microstates, the number of classes of states that we can determine from this. So quite remarkably, the MAP kinase drug complex, we get about 10 microstates from this, whereas the other complex is about 5. And the key sort of take-home message from this talk is that we can get these things called free energy landscapes, the ligand protein interaction from this. And what we can show here is that for one of the proteins, the free energy difference between the intermediate state and the final protein state is minus 3.3 kcal per mole, whereas for the other protein, the free energy difference between the least bound state and the most bound state is minus 0.7 kcal per mole. We can also calculate what we call the free energy of association, which is just the free energy of forming kind of the most loosely bound state. And the formation of the loosely bound state is about minus 5 kcal per mole for this protein and minus 5.8 kcal for this protein. So it's almost the same. So binding to the surface happens with about equal free energy in both of these proteins. But the big difference between these proteins is how deep this free energy landscape is. So it's much deeper in this protein, and this is why this has a bigger affinity. It's much shallower in this protein. It's only minus 0.7 kcal per mole, and that's why that's a lower affinity. So what we're hoping is this method could be used in drug discovery sometime in the future, perhaps. Just to wrap up quickly, quinazoline-based drugs are fluorescent and environmentally sensitive. The red edge excitation shift is significant in some of the kinase inhibitor complexes that we've tested, and we've only tested 2. The theoretical model suggests that a quadratic free energy surface is appropriate to describe these systems. the free energy for surface association, just forming a surface complex, is similar between those two proteins and the drug that we tested, AG1478. However, the main thing is it's the free energy for intrusion from the surface state, the surface state to the most deeply buried state that's different in those two kinases. And the results suggest that REES is a powerful tool for examining intermediate states in protein ligand interactions. So thank you very much. Terima kasih.
Thank you very much, Andrew. Very interesting talk, and it's exactly 30 minutes, the time that you used. So for everyone who wants to ask questions to know more about the Mr. Andrew Clayton research, you can just type your question in the chat box, and we will later discuss with Professor Andrew after the second speaker. So yeah, now I will, thank you, Andrew. So stay with us because we have panel session after the second speaker. And so I invite the second speaker, Professor Terry Mart from Universitas Indonesia. Professor Terry?
Yes.
Oh, yes. Okay. Thank you very much. So a brief introduction for Professor Terry. Professor Terry is from Universitas Indonesia, and please bear with me, Professor Terry. And Professor Terry is an expert in physics. And Professor Terry finished his education in Germany, and he also a very reputable author in count photo production, hadronic structures, and also neutron stars. Okay, time is yours, Professor Terry.
So I thank you very much, okay, I'm trying to share the screen, yeah?
Yes, please.
Can I do that? Okay.
Yeah.
So can you see the screen, I mean, in the slides?
Yes, now it is visible to us, yes.
Yeah, thank you very much. Good morning, very good morning, ladies and gentlemen. Salam, greetings from Depok. Thank you to the organizing committee for inviting me. I'm really sorry, this is a kind of talk with many formulas, mathematical formulas. So I think what I should say is just enjoy. Okay, the topic of my talk is actually a summary of my research for almost 35 years. The topic is electromagnetic production of kaon theory and applications. This is a kind of summary talk actually, but I'm really sorry that I could not present the latest result, which were published in 2021, because as you can see here, I have already almost 50 slides. I think I have no time to go to those slides, all of these slides. I will skip less important slides. So let me start with the motivation, the contents of my talk. First, I would say the motivation. I will say the motivation why kaon photo and electroproduction is interesting for us, and I will present the previous models and their problems, and I will also go to the investigation of the kaon photo production near threshold. Threshold is a very low energy needed for producing kaon and hyperon. And also I will present some phenomenological application of this model or this reaction, and I will go to the photo production models up to 3 GeV, which is almost for us is somewhat higher energies, because nowadays Jefferson Lab in United States is already using 12 GeV electron beams. So I will also present the partial WIPS model and covariant isobar models, which we were developing in the last decades or in the last years. And for the works is electroproduction, because last year I was contacted by one of my colleagues from Jefferson Lab. They have now new data for electroproduction, which are very also interesting. So motivation why can photo and electroproduction is interesting are interesting for us. Can photo or electroproduction is a particle reaction, which is we can have this particular reaction if we are bombarding the nucleon by photon beams or gamma beams. The nucleons are the content of the nucleus. So there are many outputs or many final states, particles, but in this presentation, we are mostly interested in the final state that consists of a kaon and hyperon. Kaon is a neutral kaon and charge kaon, K plus, and hyperon. We have a lambda hyperon, sigma 0 hyperon, sigma plus, and sigma minus, which are described or tabulated in the so-called particle data book, which is considered as a Bible of the particle physicists. Now, in the last 20 years, we have a very abundant data from several laboratories like Jefferson Lab in Virginia and Spring 8 in Osaka, Mainz, MAMI, Mainzer Mikrotron in Mainz, Germany. Also in Bonn, we have also experiment there, and also from ESRF in France. And well, we have also experiment in Tohoku in Japan. Now, this reaction involves strangeness explicitly, namely we have strange quark inside the particle kaon and hyperon. This is very different from the well-known particle reaction that involves pion and eta mesons. Now, what we are interested in here, or what we are doing, is we are constructing the model to describe this reaction, because we have already abundant experimental data. So this elementary operator is required or can be applied for studying the nucleon and kaon and hyperon resonances, which are the topics of resonance, nucleon resonance communities, and the production of kaon on deuteron, and also production of hypernuclei. I will tell you about hypernuclei after this. And we can also study strong coupling constant, isospin symmetry, strangeness process, which are the topics of particle physics, because the last two topics are the topics of nuclear physics. We can also go to Gerasimov ensemble, etc. But after that, we realized that we have to improve our model, because the model is no longer accurate due to the coming of the new data. So this is the example of the application. You can see in the left figures that we produce hypertriton. Hypertriton is 3H lambda in the final states, and the initial state at the bottom is helium-3. So helium-3 is conventional nuclei. So this belongs to conventional nuclear physics. And in the final state, we have hypertriton. This is the hypernucleus states. This is the business of the hypernuclear physicists. So if we want to produce this hypertriton, of course, we need a so-called elementary operator, which is described or which is drawn by this yellow ball or yellow circle or oval. So if I ignore the two nucleons in this helium-3, we have the elementary operator, which is our really our business in this topic. So in this 35 years research, I'm really involved in this elementary reaction. We constructed the elementary operator to describe the data. So how to construct the isobar model, or this model to produce kaon and hyperon? The easiest or the simplest model is the model which we constructed more than 20 years ago, because this is very simple. So this can be, or this model is reproducible everywhere. So why it is simple? Because we have only this Feynman diagrams. So I'm really sorry to those of you who are not interested, who are not familiar with this diagram, but if you are interested in, you can take a look in the standard textbook like Bjorken and Rella, Relativistic Quantum Mechanics. Now, we have nucleon resonance here in the diagram A. We have hyperon and hyperon resonance in principle in diagram B, also kaon and kaon resonance in the diagram C, and in diagram D we have the so-called contact term to maintain the gauge invariant. Now in this model, about 20 years ago, I found that we need to include a D13 1895 resonance if we want to describe the data. This is how we calculate the model. So this is, I think, very mathematics. So let me explain the result. This is 22 years ago. These are data from, I think, for all data before 1998. So this is data from 60s or 70s. So with this data, you can draw every line you like, straight line or increasing line or decreasing line, and etc. So we construct the simplest model, and we have this this result, which is plotted here as in the blue line. So if we now in 1998, Sapir collaboration reveals new data. This is the data are in this red circles. So you see that at W around 1.9 GP there is a structure, and at the time really we were thinking very hard to describe the structure, but finally we obtained that if we include D13 1895, we could explain this data. Of course, we are very happy at the time, because this is the first publication for me when I have arrived in Indonesia after my study in Germany. So we claim that we found new data and this new resonance. I'm really sorry, new resonance. And this resonance is really or fits the quark model predictions due to these parameters. The parameters are in good agreement. So finally, they put the data, the particles in the particle data book. Although they say that we do not use the following data for average, etc., we were very happy at the time because they consider our finding. So a few years later, one of my colleagues told me that, well, you are confused. That should not. It should not be D13. It should be P13. There are at least 3 colleagues of mine told me about this. So we reply the studies. I mean, we we did more study in this case. We put more data, and finally we found that the P13 actually is important. Now, the model, this 20 years ago, is put in MADE homepage. They call it a KEON MADE in Germany, because at the time the internet connection in University of Indonesia is not so good. Nowadays, I would like to move this page to my office, University of Indonesia, but I'm still very busy. So I hope that I can move this page also to Indonesia, or copy this page also. But as I told you, we have many problems. More data available, and we have also problem of that data discrepancy. This is very confusing, because as phenomenologists, we really rely on the data. So, for example, if you use take a look figure on the right, yeah, we have data discrepancy here. Sapir 2004 and Sapir 1998 are in a good agreement, but the class data are higher, or larger at W more than 1.8 GP. So if we fit the class data, of course we could do that, but we have different parameters than if we fitted the Sapir data. So this problem, I think, is not solved until now, but people tend to use class data because more and more data are consistent with the class data. So we use the class data nowadays. So this is what I told you before, that P13 should replace the D13, and I think they put it now in particle data book also. I would move to the intrinsic problem of count photo production. I hope I still have 5 or 10 minutes more. Count photo production need very high energy to create the KEON and lambda in final states. We call it threshold energy. So the threshold energy are too high for the nuclear physicists. At these energies, too many nucleon, hyperon, and KEON resonances are excited or are involved. So the models, the theoretical models or phenomenological models are very complicated. This is not the case in the pion and eta photoproduction. And then almost all coupling constants are not known by people on this planet. So this is also another complication. And we neglect higher order corrections, which is very sacral for the people in the high energy communities. And okay, there are more and more fundamental problems, but usually nuclear physicists can ignore that. So what is the solution? Okay, let's limit the energy of interest, and we can decrease the number of Feynman diagrams, and we can use also very simple formulation, very simple parameterization, like what I have written on the right side. So yeah, we made this model, very simple model, and this is the result. As you can see that the data or the energy is only about 50 MeV above the threshold. So threshold is around 1.9 GP. So this is very low energy. So, but okay, we can prove something, and also we could predict the data without fitting the data very well, as you can see on the right side. Yeah. Also, I think I will skip this because I have not so much time. I would like to present a few application of this. Yeah, I'm really sorry when I talk about application. This is not everyday application in our everyday life, because the application is also in the theoretical and the experiment or in the theoretical side of particle and nuclear physics. So first application is the investigation of neutral chaos form factors. Different from neutral pion form factor, you see that neutron, although neutral, neutron consists of 3 quarks that contains charge. Yeah. But this is also in the case of K0, a neutral kaon. Although the net charge is 0, the charge of quark that creates this K0 is not 0. So the strange quark and the anti-down quark are moving, creating a kind of charge distribution inside the K0, but the net charge outside this K0 is 0 or neutral. Yeah. Now we have some prediction, some models for this charge distribution. If we plot this charge distribution, which is actually represented by the K0 form factor, this is a kind of Fourier transformation of the charge distribution against the Q square. This is a momentum transfer of the electron. You see that there are at least 2 models. There are more actually that we can compare with the neutron form factor. So from this graphics, from this figure, you can imagine that inside these neutral particles, really there is charge distribution. So we put this model we put this prediction in our model in this Feynman diagram, and we predict that there are really differences if we use different models. So this is a sign that we could actually probe this theoretical prediction by using our calculation. Now this can be measured by experiment, for example, in Jefferson Lab or in MAMI in Mainz, Germany. So we have published it, and another application is the study or the investigation of the narrow resonance. Narrow resonance is a resonance predicted by the by Diakonov and Petrov a long time ago. Yeah, this is almost 20 years ago. After the pentaquark was very well known at the time, but it is announced by particle book does not exist. So there was actually a kind of rumor at the time, not rumor. I think there are almost 1,000 papers at the time describing or trying to reveal this theta plus or pentaquark particles. And after some measurements dedicated really for this theta plus production, people decided, and also this particle data book also decided, that pentaquark does not exist. It's pity actually, because I have published a paper on this prediction of the theta plus photo production. But okay, if this does not exist, it is well, it is this narrow resonance also probably the best candidate we can study. We have published this result in 2011, so more than 10 years ago. I can summarize that we see a clear minimum. Yeah, we see that there is a sign of this pentaquark, this narrow resonance. I'm really sorry, and energies around 1.65 GeV around here. So because at that energies, the data really made a kind of this a jump, go down and go up quickly. This happens in all angular Keon angle. I mean, in the experiment. So we have continued our models, our investigation to higher energies, to 3 GeV, around 3 GeV. We have published the result. I have no time to explain them all here, but I'm really sorry that I didn't write the last publication was in 2021, where we have actually added more and more resonances. As you can see here, this is a very busy figures. We have more and more data. Yeah. So approximately 8,000 data points for K sigma and around 9,000 data points for K lambda, and we have more. We have 1,000 data now from Jefferson Lab, which have not yet been analyzed. So finally, this is the last topic. Is not this one. We have added also several higher resonance nuclear resonances. I think this is the very good topic. We have tried to investigate the pure spin propagator because theoretically in the propagator independent diagrams, if you use higher spin propagator, there is a problem which is called the background problem. So, for example, if we have spin 3/2, we have the backgrounds of spin 1/2. So, to tackle or to handle this problem or to solve this problem, we should use pure spin propagators. But this is very complicated. But we succeeded to use this pure spin propagator in a very simple reaction. Actually, this is only toy models. We have published in 2017 and also 2019. And also we this study is not yet completed because we have just published it in the conference proceeding. So this is my last slide, which shows you that we have a new data from Jefferson Lab. We have thousands data points here, but this is very not difficult, not complicated, but consuming very much time, because for each data points we have to integrate, because the experimentalists average one datum every data points here. They average in the cosine theta. Theta is the Keon angle, and also energies. So we have only here Q square, W, or cosine theta separately as a pre-parameter. So let me conclude. I hope that you have enjoyed this talk. We have improved our isobar model for photo production and electro production, actually to 3 GeV for all isospin channel. We have obtained nice agreement with experimental data, and we have applied the models to investigate some phenomenological applications, and we have also addressed the use of pure spin representation, and we are ready actually to publish the electro production result. So I thank you very much for your patience. Thank you.
Thank you very much, Professor Terry. So yeah, it's a we can see from your work that it's very important to read the data and then to fit it with the most fittest model. Okay, so now we come to discussion session. So here in the chat box, there's no questions yet. So I invite from all the participants to just raise your hands. Oh, okay, we have Ibu Ana Safitri. Please, Bu Ana.
Still mute. Am I?
Yeah, still mute, Bu Ana.
Okay, sorry for previously because the host will allow me to unmute, not allow me to unmute myself. Okay, thank you, Ibu Zupet, as our moderator. I have question to Professor Andrew Clayton. For Professor Andrew Clayton.
Okay.
Okay, this is related to your presentation. So the first question here, are there any specific requirements for fluorophores that can show REES phenomena?
Oh, good. That's an excellent question. So what's required usually is that there's a dipole moment difference between the ground state and the excited state. What that really effectively means is that if you put your test fluorophore in solvents of different polarity, you should see a very big shift in the emission when you go from, say, a non-polar solvent to a more polar solvent. If it's very sensitive to polarity, then this would be a good Reese probe.
Okay. Okay, maybe can I continue to the next question if it's still allowed?
Yes, yes, please, Ibu Ana.
Okay, the next one, maybe how to maybe quickly convert the RES data to we get free energy to obtain a dissociation constant.
So, so that's a really good question too. So, if measuring the binding for the total dissociation constant, you have to measure that sort of separately. The Reese only tells you the free energy difference between the most bound state and the least bound state. It won't tell you anything about the equilibrium between bound and free.
Okay.
So it can't really be used for measuring the equilibrium binding. To do the equilibrium binding, you have to measure the concentration of free and you have to measure the concentration of bound.
Okay. Okay,
Yeah.
thank you very much for your answers.
My pleasure.
Okay, thank you very much, Professor Clayton. And we have another questions from Bapak Lukman Hakim, please.
All right, thank you very much. So I would like to direct my question to Professor Terry Mart. Professor Terry Mart, sorry, I think I missed some point in your last slide when you mentioned the difficulties in integrating lots of points on your data. I'm sorry, I didn't catch which part is difficult, because on the left side of the equation you show us a rather simple graphic of the containing a lot of points, but I couldn't catch which part is difficult in that slide. I'm sorry if I missed the most important part. Okay, thank you.
Yeah, thank you very much, Pak Lukman Hakim. I thank you for the question. Like I told you, this is not difficult or this is not complicated. It is time consuming, because every data point, for each data point, we have to integrate. Now, if we have thousands data points and we have to fit it, fitting process usually making a loop, 1,000 loop. So one data point we have to integrate. So it takes maybe a few milliseconds. But if we have 1,000 data points and then we have to fit it, we have to make a loop. Yeah, this is a fitting loop for also 1,000, 100,000 sometimes. So it is really time consuming. Yeah, without this integration, usually by using this 32 cores. This is from I have this processor, not Intel. What is this? Another competitor of this Intel.
AMD.
IMD, yeah.
Like Threadripper.
Yeah. This is only Threadripper, yeah. It's
Okay.
64
Yeah, that's a new one.
thread actually. So we have put this in the whole course. The temperatures of computer are increasing, of course, and it took usually one week or something like that.
Okay.
Now, if we increase this with this integration, I think we are multiplying the problem with a factor of thousands, not 1,000, but thousands, I think.
Okay.
So I'm really excited with this, yeah. But I haven't fit the data. I have just reproduced. For example, may I share the screen again, yeah? So maybe wait a minute. We need patience. Full screen mode. Okay. Okay, this one. So you can see that, yeah. So this red curve actually is our old model. This is Keon made, and you know that to obtain this curve really took time, and also it was during the pandemic. Few months or last year actually, I plotted this red curve by using my PC at home because I could not reach my office during the pandemic. So, but this is very challenging. I think very challenging. Actually, I mean, from the very simple calculation, I need 1,000 weeks actually to but I think I could manage to solve the problem.
Okay, so just let me try to clarify one more thing, that the blue dot that you show is actually an experimental data from given by that sorry, the Karman or something like that.
Yeah, Karman Jefferson.
Okay,
that's true.
Okay. Okay, so and the red curve is something that you obtain by integrating all those experimental data.
No, integrating the models.
Including the models, okay.
The model, the result of the model is integrated for each point. And you see that the data, of course, the distance between data, of course, large, but to obtain a line, the distance between points, of course, smaller. Yeah.
Yeah, right. So I misunderstood. So I thought that the blue, I thought that the blue was input for your equation, but that's not an input.
Not yet.
Not an input. Okay. Thank you very much.
I thank you. 1,000. Thank you.
Thank you very much.
Thank you, Pak Lukman and Professor Terry, for the discussion. Is there any questions for the participants? Okay, so maybe I'd like to ask Andrew. So Andrew, in our department, we usually work with extracts of plants or extracts from certain animals which has a biological activity. So do you think if we use extract, can we actually still see risk phenomenon from the extract which maybe has a natural fluorophore inside it, or do we have to control it to be more not very various environment, because in extract we have a lot of compounds inside? Is there any insight for that? Thank you, Andrew.
Thank you, Zubaida, for your question. Yeah, that's a very good point, because I guess you could imagine, let's suppose you had an extract that was fluorescent and its fluorescence was responsive to the environment. I guess you could see the red edge excitation shift phenomenon, which would be interesting, and it would mean that it's bound to a different set of environments, and those environments that are fairly restricted in their motion. Now, the problem is, and I think you've identified this in the question, that the problem would be, is it because, say, compound A is binding to compound B, C, D, E, and F, or is it, and they each have their different absorption spectra and a different emission spectra, or is it because compound A is just binding to protein B and it's in a range of environments within protein B. So I think you can't distinguish between those two possibilities with Reese alone. What you would have to do is try to purify the extract and then rerun the experiment on the purified sample, or if you're separating the extract with some sort of chromatography, you could do Reese on the separate bands, and that maybe you would see something different. So yeah, the Reese itself, that's right, can't distinguish between environmental heterogeneity that might be because a probe is in different environments in one protein, as distinct from it's just binding to different proteins. That's correct.
Okay, thank you very much, Andrew, for your answer. So I think, it's really important for us here because we develop many extracts from the plants, like specific plants from Indonesia. So we would like to know more, maybe the cellular behavior, like how is this compound actually get into the cells, how it interact with the membrane of the cells, and etc. So I think your technique might very useful for us, but we need to know more about how to apply this method itself. Okay, so thank you very much, Professor Terry, and also Professor Andrew, because the time is up now. We have only 10 minutes for this discussion. So once again, on behalf of the committee of the BaSiC, we really appreciate and thankful to Professor Terry Mart and Andrew Clayton, who has given a very interesting talk this morning. Okay, so let us bring back the forum to Ibu Childa, and thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Well, thank you very much, Ibu Zubaidah Ningsih, for bringing the plenary session 3 on time. And distinguished speakers, all participants, ladies and gentlemen, that is the end of the plenary session 3. And before we continue to our next session, plenary session 4, we will have a coffee break. But before that, let us take a group photo in this Zoom meeting. The committee will take your photo. Okay, may I count? Okay, the committee, have you ready? I will count 1, 2, 3. Well, thank you. Maybe next page. Okay, finish. Mas Aul? Is that finished? Okay, thank you. Well, everybody, kindly enjoy the coffee break. We will continue to the plenary session 4 at 10 p.m. at 10 a.m. sharp. Thank you very much. Ibu Nurjana, are you ready?
Yeah, it's
Oh,
okay.
okay. Yeah. Distinguished participants, ladies and gentlemen, we will commence the plenary session 4. Can we start right now, Ibu Nurjana?
Yes, Mbak.
Oh, okay. Yes. Thank
Can
you.
you hear my voice clearly?
Yeah, I
Oh,
can
okay.
hear your voice clearly. Well, okay. Ladies and gentlemen, we will commence the plenary session 4. For the plenary session 4, we will have Professor Muhammad Sasmito Jati and also Associate Professor Dr. Aji Ahmad Rinaldo Fernandez. Both are from Brawijaya University. And the moderator for the plenary session 4 is Ibu Nurjana. Time and screen are yours now. Thank you.
Yeah, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Welcome again to the 10 Basic Science International Conference. You are currently in the plenary session 4 of the conference, which is the last one, scheduled for about 1 hour and 10 minutes ahead. So we start sharply at 10 and we will conclude at 11:10 Indonesian Western Time. First of all, I would like to greet our exceptional 2 keynote speakers in this session, Professor Dr. Sasmito Jati and Associate Professor Dr. Aji Ahmad Rinaldo Fernandez, both from Universitas Brawijaya. Good morning, Pak Sas, Pak Aji.
Good morning.
Okay, and I also would like to greet all the participants, maybe the head of department that joining us today, and of course, all our distinguished presenter and participant in this conference. My name is Nurjana, and I will be the moderator for this session. As moderator, I will be monitoring the time. So we have about 30 minutes for each keynote speaker and 10 minutes discussion at the end. I will also be monitoring the question in the Zoom chat box. So please feel free to drop your question in the chat box during the presentation, or you can also ask directly to the speaker later on in the discussion session for about 10 minutes at the end. So, okay, let's get started. It's my pleasure to introduce our first keynote speaker in this session, Professor Dr. Insinyur Muhammad Sasmito Jati, MS, IPU, ASEAN, hopefully I spelled correct. I'd like to read a brief introduction to our first keynote speaker. Professor Sasmito Jati currently serves as Rector for Planning and Collaboration, and he also serves as our current acting dean for Faculty of Science, Universitas Brawijaya. His research interest is in biotechnology and immunology. Brief of education background, he got his master's degree from Universitas Pajajaran with the degree Master of Science of Animal Physiology. He got his doctor from Bogor Agricultural University with title Doctor of Reproduction and Biotechnology, and another Doctor of Biotechnology from Okayama, Japan. Wow. We have a very good track record of publication as well with Age Index 6 and about 37 documents that we found listed on Scopus. And a very interesting award. I think this is 3 out of many other awards. Excellent Awards in 2010 from Okayama Muslim Student Association, and then RUT Awards as Best Indonesian Researcher in Biotechnology in 2003 from Ministry of Research and Technology. This is a very outstanding award. And Peace, Happiness, and Prosperity Award as Indonesian Young Investigator in 1995. I'm still in high school at this time. So it's a very outstanding record from Professor Sasmito Jati. Okay, so I will now turn the session over to Professor Sasmito Jati to begin his presentation. Please, you have about 30 minutes.
Okay, thank you, Bu Nurjana. May I share my presentation in PPT?
Yes, please.
Okay. Is it clear?
Yes.
Sorry. Is it clear? my PPT?
Yes.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
My topic here today, it's quite often we talk about this one. The title is Biological Fingerprint Expression of Sauropus, Androgynous, and Elephanthropus caber in terms of immunology pathway as functional foods. Of course, my team is Ika Kristina, Yuyun Ika Kristina, and also Dinia Twijayanti is our assistant of professor. Okay, this is the background of our research. As all of us knows, Indonesia is flora mega diversity. It's very important for us. Indonesia consisting many varieties of tropical plants. Indonesia is the second largest biodiversity in the world. There are about more than 28,000 species of flowering plants, and Indonesia consisting more than 2,500 different kind of orchid, and also more than 6,000 traditional medicinal plants used as jamu. Maybe all of you knows jamu is Indonesian traditional drug, and it is a notice for me. It is very important thing that herbal drugs now growing popularity. You can imagine that from $62 billion in 2005 and now become 5 trillion US dollar in 2020. So you can imagine this popularity of herbal medicine. But to understand about drugs, that means we need some require for masses. What we call it 3A. The first is accessibility, and then affordable, and also assure safety. It is something in basically important for us to understand about that. Do they fulfill the requirements? That's the first question. If we try to implementing or to use herbal medicine, accessible, of course, for Indonesian, more than 50% Indonesian population depend on alternative system of medicine. So it is a very popular in Indonesia, moreover in long time ago, but now in near future become more popular again. How about affordable? Herbal drugs are cheaper than generic drugs, so you can imagine that. In our traditional housing, you know, the toga herbal, that's mean almost all Indonesian in rural area, they have housing in the backyard. They grow some traditional plant that support herbal medicine. So, but no, we should think about that again. How about the assured safety? Of course, herbal drugs have been used in Indonesia for more than 1,000 years. But until now, scientifically, it's not have clearly understood. So it's very important to us how to manage why they use this herb or traditional. So I will explain. Follow, yes. And then one thing that I want to say, the plant fingerprint is very important way of thinking, because we know thousands of plants spread to all of tropical Asian country, tropical Southeast Asian country, but in certain case, this not express what the chemical compound that they want to. So that's why I just want to say in a plant, in every plant, in every living material should be have DNA, and DNA it's blueprint of life or their life. But the important thing that I want to say today, every living thing, what we call it, we have fingerprints. You can imagine this finger. Our finger have a certain pattern, and every pattern, every ethnicity, if we think about human, have a special in different pattern. And every pattern have essential meaning. It is very important for us to understand about that. Of course, today I do not think about DNA itself, but for us, it's important saying DNA and expression of DNA. If we think about expression, that's mean you know about the functional and express of the every plant that have species, the species variant have specific expression is very important. But the expression itself, if we know about the expression, protein expression, that's mean even in the same variants, in the same species, but if you grow, if the plant is growth in different place, different soil, also some different expression. It is very important to be noticed, to develop the strategy of herbal medicine. Then we not just saying about continuity from plant to drugs. This is very important way of thinking also to discuss about. The first step, all of us knows plant and food, and then functional food, and then herbal drugs, or we call it jamu, and then low molecular weight drugs. So in this term, we today discuss about in this area. Yeah, in our research now, I will tell you, I will presenting this discussion about the herbal or functional food. Jamu in Indonesia herbal medicine, according to the National Social Economic Survey 2021, more than I have told you before, more than 50% of Indonesians treat their body without medical help, but they are choose traditional medicine. Jamu as a traditional herbal medicine that have been used by the Indonesian community for several years. And jamu is made from the part of plant, leaf, roots, fruits, or animal extract, which has the active compound to healing a certain disease. And if we think about the functional food, it's very important to us. The list 4 categories of functional food as defined by Academy of Nutrition and Dietetics. There are 4 types functional food proposed by American Dietetic Association, ADA. The functional food are conventional foods, special dietary food, enrichment food, and medical food. That's very important one. If you look this picture, it is very important. This is Indonesian, yeah. This is Indonesian food, and all of us have been eaten almost daily. If you look this, no, every Indonesian traditional food, so many spice there. And if you know about the spice, every say that's meaningful for traditional medicine. So our traditional food is a such kind of functional food, but unfortunately, now our country, our people have invaded with westernized food, that what we call it junk food. No, junk food also become more popular, and I'm worried about that. The 4 functional food that I have told before, of conventional food, medicinal food, and modified foods, special dietary food. In this occasion, I just think about the special dietary pregnancy food. You know, our beloved mother, they also have a special food that we should make them happy, not only from our mother, but also the baby. Because we think about our future generation, we should think about our mother and also our baby. Yeah. In terms of pregnancy, as you know, the have higher risk and are more susceptible to infectious disease. So that's why it's very important thing to do. This unique immunological condition and also the spirit in the need of the amount of extract of iron. For example, iron is needed for the development of plasma, placenta, and iron transfer to the fetus. So that is very important. Sometimes the mother is not enough to eat the iron, for example. And then also, if you know the mechanism, a unique mechanism of immune system in pregnancy, you can imagine that the baby of what we call it, the small baby and very small baby, we call it embryo. Embryo attached to the uterus or endometrial part. Yeah, embryo is what we call it foreign protein. When attached to the mothers, you can imagine this immune system will work. But as you know, the mother is still healthy. So it is very important thing. The anatomical barrier between baby and mother through spirit circulatory system within the placenta, it is very important thing. That's mean embryo like antigen. So sometimes there's some unique mechanism, what we call it major histocompatibility or MECA antigen experience to is reduced to trophoblast on the fetal side of the placenta. So develop of an immunosuppressive environment with the uterus is very, very important to understand about that. And if we know about the immune system during pregnancy with bacterial infection, we try to make infection with two kind of bacteria. Yeah. So you can look this pattern. It's very interesting pattern here. Yeah, you can look this pattern. This is a phenomenon that the increasing of certain hormone support to pregnancy. And at the same time, if there is some bacteria come, so it is what we call it, the mother is susceptible to infection, yeah, including salmonella and also Escherichia coli. So it is dangerous for mother, also for the baby. You can take this one. Yeah. So if we think about that, we should do with accessible, affordable, and safety something to help the mother with our ideal condition. So we try to combine two kinds. It is our experimental. L4 scaber in Indonesia, we call it tapak liman, and also Sarapus androgynous, we call it in Indonesia daun katuk. It's very famous and very easy growth in anywhere in Southeast Asian country. But as I have told before, think about the fingerprint. Even this is possible to grow anywhere, but not all this plant will express a certain fingerprint. That's why the environment surrounded the plant and also soil, it is a very important thing. So that's why I take it this two plant from surrounding Malang city. Yeah. And then if we think about the Salmonella typhi, typhoid fever symptom like high fever, headache, weakness, dry cough, and stomach pain, constipation, also raises, and Escherichia coli like a diarrhea, abdominal cramp, bloody orange, and also lost appetite. And this is my research result. What we call it, phytochemical fingerprint of Elephantanthropus scaber L. From the HPLC chromatogram research, we can look this, that I already make circle, red circle. This is expression of fingerprint special for Elephantanthropus scaber. Yeah, you can look this one. And then all of them, we can look this one. And we already published this expression in this year, in the early of this year. That's why Elephantanthropus is very potential plant to support what we get, because they express triponate compound from ethanolic leaves extract, LCRMS, also LCHRMS chromatogram to display with six spectra of triponate compound, olenate acid, agar-triol, and also caripelline oxide, deoxylipontimine, also scabertropin, and also ferricarol. Another expression of Sarcopus androgynous, you can look the expression with what we call it fingerprint also. Even in this occasion, this is not my own research, we just take from Zheng et al. They express fingerprinting for Sarcopus androgynous. And why we combine this extract? The first, the logical way of thinking of multi-target approach that we want. Combination of herb are normal. Yeah. Empirical observation and reasoning based on a particular patient. Also, this herbal combination may not be targeted to a particular organ or multi-targeted cell, or tissue, or any biochemical system, making his synergy even more difficult to identify possibility of drug interaction, and also adverse reaction arising out of this have to be checked before coming out with the drugs. And also we think about the synergy system. Most of the effective of phytomedicinal in the market are as whole extract of the plant. So it's very important thing. But practitioners believe that synergetic interaction between the component of individual of mixture of the herb are vital part of therapeutic efficacy. And mechanism and action of many phytomedicine is still unknown, of course. There are several instances where a total extract show better activity than an equivalent dose of isolated compound, chemical, and pharmacological evidence to demonstrate conclusively the concept of synergism. So this is what we call it way of thinking of balancing system in every mechanism in our body. You can look this one, this just logical way of thinking. You know, this one is cytokine, yeah, cytokine, yeah, like interleukin 4, interleukin 5, and other things. Interleukin 10, TGF beta. That's not just increase or decrease, because we call it a modulator. That means sometimes increase, sometimes decrease, sometimes stimulate, sometimes inhibit it. This way of thinking in biology, in physiology also. So in our research, we try to look this one. We do some experiment, animal preparation, extraction process, also bacterial infection by orally. We make some treatment like this. Yeah. The first, E. coli treated with 100% of Elephantanthropus scaber, or the formula is only Elephanthropus scaber. Another formula, 75% Elephanthropus or ES, and 25% SA, Sarcopus androgynous. And then another formula is 50-50. Another formula is the opposite of that formula. And we try to see by facts analysis. And also we know the safety or not, the negative effect of not. We look hepar and kidney histology. This is very important, like what we say about the negative effect. So it's very interesting when I look this one. Not just how to say cytokine increasing something and also some stable something. Also, we can look this one is very important thing that we want to see, like this hormonal. Yeah, this hormonal for mother hormone also like increase prolactin is make breasts become high, become bigger. Sorry, and then they make some milk production. It's very important for us. At the pregnancy period, C4 show the high level of prolactin, also estradiol and others. Yes, and if we make combination, also it is interesting facts that we look some important thing here. FS8 level and so LH level, and we already published this result, and it's very interesting for us. And so we can look the peak of how to say here, our during bacterial infection, the bone marrow accelerated to produce granulocyte, support a rapid recruitment of this into the blood circulation. E. coli infection also stimulate proliferation of granulocyte precursor, make the high production of immature GR10 or granulocyte cell in the bone marrow. ES alone also combined with ES or combined with SA ratio or something like trigger the relation about the mature granulocyte from the bone marrow. Okay.
5 more minutes.
Yeah. 5 more minutes. I will try to make this another reason. After we make some, should be understood. Every plant have compound that's possible become toxic. So dose is very important for us. That's why we check liver and kidney. In our dose that we have make formula, it is safety, like what we see, affordable, and then also safety, and then also accessible. So in our formula, we can guarantee that this formula is safety also. And then another thing, the combination of SA and ES accelerated the production erythrocyte progenitor. This is very, very interesting, what we call infants, because if we think about the TR119 and CD4, that's mean this progenitor cell will support many kind of metabolism. This is very important thing. Even we have not known yet in detail what is the mechanism will supported by this cell, but it is indication SA and ES combination accelerate this production. It's very important also. Also, EL2 production on CD4 cell activation in typhoid pregnant mice. Yeah. Also another thing, like combination SA in promote TNF alpha, decreasing interleukin 4 production in the typhoid pregnant mice. I try to make more quickly. I want to say about this combination. The first, if we think about the immunity, the first step of our defense is what we call it innate immunity. Yeah. The first step of our defense mechanism, and then the second step is adaptive immunity. When the infectious disease come to our body, the first defense system is granulocyte, monocyte, and then inflammation. If it is long term, it's not finished, it will adaptive immunity will work. So the combining both extract could activate innate immune system by accelerating the maturation and release granules of GR1 and bone marrow to eliminate bacterial infection in the systemic circulation. This herbal formula also promote interleukin-2 production, resulting in increasing level of CD4 cell in interferon gamma production. I will make summary. It's very important thing that we should understand about that. I just try to skip another result of research. The first, if we think about immunomodulator, antioxidant hormonal action, and also homeostatic control mechanism, you can look in this our research. It is very interesting one. This is a formula that we have shown before. That's make immunomodulator become balanced. That means if we think about balancing system, that means it is healthy. And also think about the, we think about, I make, it's the wrong touch. Yeah. Yeah. And also antioxidant, yeah, this is our formula. To trigger TNF alpha and also interleukin-2, also interleukin-4 for hormone action also, progesterone, FSH, LH, prolactin, and another thing is anti-cancer. Even we not mix so clear in this occasion because we have limited time to understand about that, and also antibacterial. We have also published this. So this two combination from SA and AS, it is have potential with the system of synergetic system and also multi-target system. I think this is enough. This is my team. My assistant is Yuyun Christina and also Dinia Dwi Jayanti. That's all. Sorry, maybe we're losing 2 minutes. Buana, thank you. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Prof. Sasingito Jati, for giving such an informative and comprehensive presentation for the first part. I will keep the question later in the discussion session. So all participants, please drop any question for Prof. Sasingito Jati in the chat box. And now, ladies and gentlemen, all the participants, we shall continue to our next speaker, the second part of plenary session 4 with Associate Professor Dr. Aji Ahmad Rinaldo Fernandez. Are you here? Oh yeah, there you go.
Yes.
Okay. Good morning, Pak Aji.
Morning.
Maybe before we start, I'd like to read a brief introduction to our second keynote speaker. Okay. Can you please form the slides, or maybe I'll do it? Okay. Okay. Our second speaker, Pak Aji, he's currently the head of computational simulation and statistics laboratory. He's currently the one of staff academic in Department of Statistics, Brawijaya University. His education background, he got her first degree in Universitas Brawijaya with the title of Sarjana Science as SE of Statistics, and then master degree from University Hasselt, Belgium, Master of Science in Statistics, and the doctor, he graduated from Institut Teknologi 10 November Indonesia, Doctor of Statistics. He have a very also good publication track. Currently have 78 documents in Scopus with H index 14. Wow. These are brief. I don't think I will manage the time to read all of this, Pak Aji, but the range of your publication is very outstanding. with all these ranging from statistics modeling in many kind of area, I think, social, economic, and several books as well. And his current research from 2021 to 2022 up to 2024, I think. These are all his current research. So I think we can later on discuss detail about his research. But for now, I'd like to give opportunity to Pak Aji. I'll turn the session over to you. You have about 25 or up to 30 minutes. I think we still have time for that. Please. Thank you.
Okay, thank you very much. Let me show the presentation. Is that clear for the presentation?
Clear.
Yeah, but
Okay.
still not on
Full screen, yeah. We need
full screen share.
for full screen.
Okay, is that clear now?
Okay,
Okay.
thank you.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, good morning, and healthy greeting to all of us, ladies and gentlemen, the Honorable BaSiC organizer and speaker, as well as the audience. Pergi berlibur ke rumah paman, beli cilok 5 ribuan. Izinkan saya untuk perkenalan agar acara ini dapat berlangsung lebih nyaman. Sorry for Indonesian language. Yellow is the color of honey. The color of the sky is blue. Hello, my name is Aji. I came here to get to know you all of you. I'm from the field of statistical modeling and data science development. On this happy morning, I will present a material applied statistical modeling and data science in nursing community post-pandemic. In statistics, there are two things that becomes the focus, namely method of data collecting and data analysis. The first part, method of data collecting. In this case, data is collected by collecting data. Nowadays, data can obtain from anywhere, data obtained from available data source. Keep in mind that data is not only in the form of numbers, but can be formed the image, sound, video, text, etc. All of that can be referred to as data as long it can be used as material to view the environment, object, event, or concept. In the field of data science, there are several ways of collecting data, including information extraction, information retrieval, natural language processing, clustering, categorization, visualization, text summarization, and so on. The second part, method of data analysis. After the data is collected, the researchers needs to determine the statistical analysis that is in accordance with the collected data and is appropriate to be able to answer the research objective. From this data, information will be obtained which can be used as support in decision making. The advantages of data science in accommodating things that cannot be captured by conventional statistical analysis are as follows: better data processing, supporting business decision, detecting transaction anomalies, and technology development for data processing. Based on this opportunity from the development of data science and base statistic, therefore, inshallah, next year, 2023, the Department of Statistics, Faculty of Science, University of Brawijaya will have a data science bachelor program, accompanying the statistic bachelor program and also statistic master program. Currently, statistical analysis that is in spotlight for some statistics users are exploratory analysis, comparative analysis, and associative analysis. The first part is exploratory. The purpose of this analysis is to extract from the data any information that can further utilize. In practice, this analysis is used as information of variables that are new variables. In this analysis, there are several statistical analyses that are part of it, namely cluster, B-plot, principal component, and factor analysis. The second part is comparative analysis. Is an analysis used to determine which difference between two or more treatment or group in one or several variables. The several analysis tests included the comparative analysis are t-test, ANOVA, MANOVA, MANCOVA, and etc. And the third part is the main focus of our discussion today, In this analysis, the focus of the research is to test the relationship between variables used. One of the most popular is structural equation modeling, or SEM analysis. SEM is divided into two models, namely structural and measurement. In the modeling, these two models require supporting theory and previous research that is used as reinforcement in establishing the relationship between the variables to be studied. In looking for the theories and research beforehand, often no previous research is found that matches the existing variables, so that the researchers find it difficult to reveal the existence of these variables. Therefore, with the existing of the data science, it can be used to form the new variables through discourse network analysis, or DNA. In DNA, it is divided into social network analysis, or SNA, and sentiment analysis. Nowadays, in the collecting and procedure of the data that is increasingly diverse, both in size and type, it requires us to be able to adapt to keep up with the existing development. Today, it is known as data science. In today's era, conventional statistics needs to be developed. Data science is one way to develop statistical modeling that can be in the term of big data. Here, the data science play a role in helping to explore the broader and more comprehensive information with real-time condition. For example, information mining, which is divided into two things, including variable mining and relationship mining. On this occasion, we will discuss a little related to one method of data collecting, namely by data mining. This method also includes information mining. Then, in mining this information will be used as support in the existing statistical analysis, structural and measurement model in SEM as main topic of this discussion. Also, today's discussion explore the flexibility modern statistical modeling analysis. Just wait a second, in several pages ahead of this presentation. Don't worry. And also, do not forget to take your cup of coffee or tea. Statistical modeling for the response variable with continuous data scale is divided into two major groups, namely structural modeling on the right side and measurement modeling on the left side, where the structural modeling is based on the linearity from each variables, while measurement modeling is based on the data scale. Development are currently underway for structural modeling with non-parametric, semi-parametric, and measurement model that are more flexible, able to accommodate mixed data scale, nominal and ordinal, accompanying interval and ratio scale data scale, as well as reflective and formative measurement models, measurement form in several studies. Statistical modeling for the response variables on categorical data scale is divided into two basic, namely statistical based and computer based. The hybrid concept that is currently being carried out is a hybrid between SVM analysis, cluster, and the latest development being carried out in additional of fuzzy concept in the two main analysis. It is also further discussion the hybrid of classical discriminant analysis and classification and regression trees. In addition, the hybrid concept that has been carried out between classical statistical modeling and computer science, such as logistic regression with boosting and bagging. What is currently being done is the analysis involving multi-group moderation variable. Analysis was carried out on each characteristic. Information mining is a procedure for collecting information for data so that valid relevant information is obtained. One implementation of information mining is variable mining and relationship mining. The flow of variable mining through text analysis start from mining information from the text, then pre-processing the data. Then research variable will be arranged based on the information obtained. The flow of variable mining through social network analysis start from mining social media information by including the most popular actors and opinions. The analysis that can use to mine large amount of information from the internet or the social media can be in the form of structures and network. For the example, web information network analysis and web community discovery, opinion mining, usage mining. The derivative of variable is obtained from the variables. Seen from the right side, the derivation of culture variable, it should be innovation culture, work culture, and moral culture. It may be obtained from the DNA, SNA, and text analysis. The leadership variables reduce the transactional leadership variables, then trans-global leadership, and Ki Hajar Dewantoro leadership. Also, again, with DNA, SNA, and text analysis, and or maybe by Fernandez leadership, of course, hopefully. Okay. An important part in establishing the relationship mining is designing the inner model, making the relationship between latent variables. In some research goal, it is usually state the formulation of the problem or research hypothesis. The basis of the designing of the model can be following 9 points. You can see from this left slide. It is possible that the relationship between latent variables can be the form of proposition. Thus, the analysis allows use to the exploratory analysis of the relationship. To get the relationship mining, which is part information mining, here the data science-based analytical tools play in a role in developing it. Some analysis that can be used in this case are text analysis, discourse network analysis, social network analysis, and so on. This modeling is adapted from our research and service grant this year, namely BRIN and Dr. Mengabdi. The following are examples of statistical modeling and data science on behavioral modeling and community perspective on stunting problem area. The following provides behavioral modeling and community perspective on stunting level before and after DNA, where there are 4 variables that are formed based on the result of DNA analysis, including health services, environment, parent nutrition status, and children's diet. And the model was also developed based on DNA mapping, which is one of the network analysis on big data source from media, mass media, social media interaction, etc. This modeling is adapted from our last year research grant. The following is an example statistical and data science modeling on community behavior modeling for the COVID-19 vaccine. The following provides a behavioral model for vaccine based on the theory of planned behavior involving attitude, subjective norm, control behavior, and intentions. The model was developed by involving the technology acceptance model, involving the usefulness and convenience of the vaccine variables obtained from the DNA mapping that vaccination cases can affect the intention to receive the vaccine and behavioral toward the vaccine. Social network analysis, or SNA, is a tool and process with the aim of knowing of relationship and network structure. An individual or group of individuals can be described as a network of individuals connected through patterns of interaction and social connection. The data obtained comes from tweet data or Twitter, a total of 2,530 tweets. Tweet data is imported using the Node.xl basic program. The keyword is used is new normal. 5 actors who have influence with other actors are a writer, authors, cabinet secretary, archaeologists, and 2 others people. SNA can be used to measure the strength of relationship formed. The individual level, there are several measures to measure the relationship, namely degree, closeness, and between centrality. Degree centrality is a degree that can see the popularity of actors in social network. The measure can see the position of the individuals in the network. If in a directed network, an actor or popular individual who is often contacted is called an independent Meanwhile, an actor or individual who often contact where the link leads out of the actor is called out degree. In the result of the analysis, Mike Duncan has the highest degree score, which means that the actor has the highest level of popularity. Between the centrality can show the position of the actor, node, and the intermediary betweenness of the relationship between actors, individual, with other actors, individuals. The centrality value possessed by the highest actor is an actor who has good liaison abilities between an actor and other actor in a network. The result of this analysis show that Mike Duncan has the highest value between centrality, which means that many actors depend on making connection or interaction with other actors. The result of SNA also obtained variables and relationship mining. Several indicators have been found from the data scraping process, and SNA can be used the measurement of the purchasing process variable in the new normal state, namely waiting time until item can be received as a customer satisfaction, risk of transaction failure due to system as a porter's force, seller response speed to customer problem as a customer satisfaction, and item purchase incentive, discount, promos as a customer engagement. After obtaining mining variables, as modeling of relationship mining can be formed that can relative variables to another, you can see from the right slide. Discourse network analysis, the second part, is a methodological approach that combines discourse analysis and social network analysis to identify a discourse in various documents to form a network. Some of the advantage of DNA that can see 5 things, which actors are involved in a discourse, relationship within each actors, relationship within each concept, and the relationship between actors and concept. The slide show that the result of DNA with the keywords in news, namely digitalization of electronic money in the new normal era. In this picture, the black circle icon is the actor, the black square is the issue, and the green line is the positive sentiment. Of course, the red line is the negative sentiment. There are several important issues regarding these keywords, namely digital transformation, public trust, regarding the security of digital e-money, ease of transaction, and others. The result of DNA can also be used to mining the variables and mining the relationship. The picture on the left show the result of DNA regarding the factor driving the Indonesia's green economic after pandemic. The result can be seen that the important issue that arise are investment, marketing, innovation, and digitalization, the use of natural material, and others. Furthermore, this issue can be linked into the variable of the model, namely digital marketing. In this picture on the right, DNA can obtain the new things or new variables that can obtain the influence relationship between digital marketing on Indonesia's green economy. Sorry for bothering you. I will skip this part because I think there's not enough for me. Okay, maybe comes to the next slide. Okay, this one. Pages. Okay. Here is the innovation product of our grant, output of the grant. Here is an innovation product that was made for one of the output from the research grant that I did. The innovation product development is an information system or software that can perform DNA analysis integrated with SEM analysis. DNA analysis can be done by entering the news or social media that has been searched for and looking at the issues or statements set by several actors. This innovation product can provide benefit for agencies or government conducting research on causal relationship between variables, As I explained early about DNA can perform variable mining without the need to look at previous research or the theory background. This, of course, can save time to look at the variables that have a high probability of influencing an issue. And also, do not forget to take your cup of coffee or tea. Okay, the following is an example of visualization of the result of relationship between actors and discourse or issues that can contain positive and negative agreement. The result of DNA, of course, especially for this our product, DNA analysis on this product can explain more detail that similar product. DNA result can show a list of actors involved in the search of particular issue from the news. In addition, other results that can be shown are a list of issues that a number of related issues. Furthermore, there are many links or relationships that contain positive or negative relationship, the number of documents and others. The summaries cannot yet be displayed on similar product. That's why this is an original product for me. Here is an example of SAM modeling step that can be done using data from questionnaire that can also be filled out in this information system or in this software. SAM modeling on this product has advantages because the data used can be directly imported from the questionnaire data that has been made on this product as well. We do not need to bother to open the Excel first, save and enter it, and in the software as usual. It's easy to use. The following is an example of result of SAM analysis in the form of loading factors, outer weight, and path coefficient. The path coefficient and the p-value obtained can show the relationship between significant or insignificant relationship variables. Basically, because the system has been integrated into a single comprehensive system, there is no need to open other tabs, which can cause the PC or laptop to be slower, so that the process from the data input and data analysis to the end can be faster. Some things that can be done after relationship mining are modeling using statistics modeling is done so that the information data generated from the mining result can be interpreted to obtain the new information. To present something, concept ideas that exist in the problem domain, as well as to present the relationship that occurs, structural modeling can be used, one of which is by using structural equation modeling or SEM. SEM can be divided into three types, namely parametric SEM, non-parametric SEM, and semi-parametric SEM. Parametric SEM has a characteristic, namely the relationship curve between variables follow a certain pattern on or form a clear data pattern for especially linear parametric. Non-parametric SEM has a characteristic, namely the relationship curve between variables is not known in the form of the pattern. The third one is the hybrid, as we call it, semi-parametric, has the characteristic of combination of parametric curve estimation and non-parametric curve estimation. Modeling that discuss the relationship between variables is commonly referred to as structural modeling. The influence on the structural model is divided into several types, namely direct, indirect, and total effect. The development of the concept of indirect and total effect in semi-parametric SEM and in its interpretation is urgently needed. In semi-parametric, the direct effect structural modeling is divided into several conditions. First is direct effect with linear parametric. As we can see on the left and the right side, direct effect divided into positive on the left and negative on the right. If beta is more than 0, then the relationship is in positive linear. If beta is less than 0, then the relationship is in negative linear. Second part is direct effect of non-parametric truncated spline linear. The direct effect is also in semi-parametric SEM. For an example of direct effect of positive non-parametric truncated spline slope on a semi-parametric model, it can be seen on figure A and figure B. with positive in both positive linear slope. The figure C and D represent the negative linear slope. And the last figure is about the two slope with different sign. The effect occurs of this relationship between exogenous and endogenous variables is mediated by variables commonly referred to as mediating variable. In semi-parametric SEM, the structural modeling of indirect effect is divided into 4 conditions. You can see with the indirect effect of parametric linear, mixed non-parametric and parametric, mixed parametric and non-parametric, and non-parametric truncated spline. Indirect effect of parametric linear is obtained by multiplying two direct effects. If the direct effects are both positive, then the indirect effect is positive. If the direct effects are both negative, then the indirect effect is positive. If the direct effect is indifferent in sign, then the indirect effect is negative. The difference between two graphs on the right side show the different strength of indirect effect in the left regime of knots, Q1, compared with the right regime of knot. The third and the fourth condition coming up these two figures with different type of indirect effect. You can see the difference between the 4 indirect effect can be a long discussion for the statistician, especially for me and for my research team. Sorry, underdeveloping. Do not forget with your cup and coffee and tea because the time is very limited. Before the warning from the moderator, after discussing direct and indirect effect, next discusses about the total effect. Unfortunately, we are not ready yet about the total effect. For further information, please contact me. A country girl loudly singing. A fairy is chopping a lime. If you need helping with anything, feel free to call me anytime. I sincerely appreciate your attention today, and that brings us to the end. Thank you so much for your interesting and attention, and also do not forget to be healthy and happy. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, thank you. Wow, that's a very interesting and insightful presentation from our both speaker. Well, ladies and gentlemen, all the participants, after we hear the two presentations, now it's time to have a discussion with our speakers. I'll start with the question for our first speaker, for Prof. Sasmito Jati first, before we continue with the second speaker. Well, I'm not from biology, but I'm a loyal customer of various kind of jamu, Pak Sas. Maybe anyone, if you have any question, because I see in the chat box all the question currently for Pak Aji. If you have any question for Prof. Sasmito Jati, please feel free to raise your hand and ask the question directly. Any? Here I see someone raising hand.
Itu Bu Ana.
Not yet.
Ana Safitri.
Bu Ana Safitri. Bu Ana Safitri, do you have any question to Pak Sas or?
Okay, yes, sorry, because I cannot unmute myself.
Oh,
Okay, I
okay.
have one question, maybe for Prof. Sasmito Jati. For the treatments to pregnant mice that infected with E. coli because the subject are pregnant mice, did you also check the condition of their offspring or their mice babies after the treatment?
Sure, yes.
Okay,
And you know, after the treated, all of the pregnant mice is different result. Of course, some of the finest formula is, I forget it, maybe 50-50 ES and also ES and SA. 50-50. These are our formula results. Yes. Can you get that point? Yeah.
Yeah. So for the combination, because you did combine the combination of maybe in Indonesia we call it tapak liman and daun katuk for the treatment. Are they more powerful if we combine them together, or maybe if just tapak liman only or daun katuk only? Is there any reason why you combine them? Are they more powerful
Yes,
for
of
the
course.
treatment?
For the result is different if we make a combination that I have taught before. Sorry, this a little bit crowded. Yes. Another place for uh, the best formula is 50-50 because as I have told to you before, tapak liman is good for in tapak liman there is what I say before imprint fingerprints is special for circulation system because there is not just fingerprint itself but also include in this tapak liman ferrum, ferrum. They support another metabolism because everything about the ferrum, in the blood system, yeah, you need the ferrum. That before I told you, some problem of women that they were pregnant is also ferrum. And another thing about the daun katu, I think it's very popular for women in pregnant. It's very popular. I know it's traditional herbal. They also support that I have taught before uh, lactation system, and then also estrogen, also FSH and LH. Yeah, these are very important to support the pregnancy. So if you try to make this formula, of course, not all mechanism we understood because it's so complicated. As you know, herbal is many things. There's whole holistic system, but in fingerprinting is very important for us. For the tapak liman is different, and then daun katu also different, but both of them support the pregnancy. Both of them. Yeah. Is that clear?
Okay. Thank you, Bu Ana.
Thank you very much. I cannot unmute myself
Pak
again.
Okay. Thank you, Bu
Thank
Ana.
you.
Yes, Prof. Sasmito, herbal drugs currently getting more popular, and maybe for some reason because it is more affordable. Sometimes it's closely correspond to the patient ideology as well. Maybe concern about the adverse or side effect of the chemical or synthetic medicine, and so on and so forth. So maybe we can give us any suggestion for us maybe to understand or raise awareness of the side effect, because I don't think that it has no side effect at all anyway. So maybe how can we aware if the herbal drugs currently in the market is safe or not? Very general, but I think we need maybe
yes, it's a very important question. It is very and fundamental question because so many herbal medicine, it have not scientific proven yet.
Exactly.
It is very, very, very important way of thinking. And as you know, as we know, in every plant has toxicity material. It's a very important. That's why we try to make formula. That's mean dose is very important. In our case of our study here, our formula is safety. We look from our experiments from HEPA and also kidney. There is no negative effect. That's why we make this formula. Of course, if we make a formulation, it's not easy. Sometimes we trial and error system. But in this case, we already make so many dose, but the final dose is like with what I have done it in our research. So in this dose, it's safety, but of course it's still in mice, not in human. Yeah, it's not in human. That's why it's still a long story to do some research.
Okay. Okay, thank you, Professor Smito Jati. I think because of the limited time, we have to continue the question for the second keynote speaker for Pak Aji. Pak Aji, you have a lot of questions. I don't think we can manage in 5 minutes, but maybe we can highlight a few question in there. Maybe any other question we can discuss later with Pak Aji. I have a lot of questions as well, actually, but let me just limit myself. The first question from Anindya Aprilianti, that's about the data science approach for statistical modeling, how to modify model based on hybrid approach in statistics term, especially in Bayesian part. I'm not sure if this one related to your research, but maybe you have any answer?
Okay. Thank you, Bu Nurjana and Bu Anindya. It's nice to see you again, Bu Anindya. After a long time, we didn't have direct discussion because we are in the same areas in ITS Surabaya, doctoral degree. First questions related to the Bayesian parts. It's easy question, but very difficult to answer because I'm not in specific Bayesian area. However, I'm currently researching with Mrs. Ani Budi Asuti. I think she's in this room now. The development of a Bayesian model in survival analysis, which is certainly very interesting. And a hybrid model can be developed in this case by first mapping the variables and their relationship, of course, with DNA, SNA, and the sentiment analysis. I think the sentiment analysis, DNA, and SNA that you asked in the second questions. Now, a few questions maybe for the further, for the future, is the use of primary data that complement secondary data. Especially survival analysis is used more secondary data. And also the DNA SNA, it should be the primary data. This will be the best solution for me. DNA, the last question, I think, DNA, the difference, yeah, what is the difference? DNA is formed from issues obtaining from the mass media, news or newspaper. SNA is the form from the issues obtained from the social media. That's the difference thing. While the sentiment analysis is an issue that is mapped into positive and negative sentiment. How to make a choose? First, try it. Second, think to do which one is the best solution for the research goal. I think that's enough for the question from Mrs. Anindya. I think there is no another question for Mrs. Anindya because there is enough 3 questions. Thank you, Bu Nurjana.
Okay, Pak Aji. I think she's not here as well. If I look at that, it's not no longer in the room.
Oh.
Maybe Pak Aji, short answer for question from Ibu Paramita Dewanti about the why you choose the spline truncated, and then I think the most important question is at point 3. How's your research build the measurement part of SEM in semi-parametric modeling?
Okay, let me try. Thank you, Mr. Paramita Dewanti. The first question is very clear. Truncated spline is one of the non-parametric part or non-parametric regression approach in the form of an explicit function, in contrast to other approach that are more implicit, such as smoothing spline that use integral as problem solving. I think that it's not easy to interpret the model with using integrals. Or the kernel approach use one or more kernel function that are not visible in the form or function explicitly. That's the first question. This also answers the second question. Semi-parametric SEM is graphically referred into the example. Okay, I think maybe the last one. The third question is related to the measurement part. For SEM parametric, confirmatory factor analysis approach is a solution in the form of reflective indicators that prioritize common factor as the character of the each indicators. On the other hand, the principal component analysis or PCA approach is a solution in the form of formative indicators that priority as mutual exclusive indicators between each indicator within each indicators. I think that's enough for me. Thank
Okay,
you.
okay, that's really clear. Unfortunately, due to the limited time, I'm sorry. Maybe any other question you can directly ask to Pak Aji, as he mentioned, you can call him anytime. He's a very busy person, but very nice one. So feel free to discuss with him for any further question. So thank you again to our exceptional, outstanding two keynote speaker in the very last session of plenary session. And thank you for the participant for joining us in this session. We hope you enjoy the remainder of the conference, as we still have parallel sessions to go until about 3 p.m. today. So I think that's all for me. Good morning. Wassalamualaikum warahmatullahi wabarakatuh. I'll give it back the time to
Waalaikumsalam warahmatullahi wabarakatuh.
Bu Gilda as Master of Ceremony. Thank you. Thank you. Bu Gilda? I'm sorry, we about 5 minutes behind the schedule. Or maybe I can give to Bu Ana Safitri to continue the session. I think the next one should be the parallel session that the participant.
Yes.
Okay.
Hello, sorry, sorry.
Yeah.
There are technical problem. Okay. Thank you very much, Ibu Nurjana, for bringing. Huh? Oh, okay. Sorry. Sorry. Thank you very much, Ibu Nurjana, for bringing the plenary session 4. Well, distinguished speaker, all participants, and ladies and gentlemen, that is the end of the plenary session 4. Now we will continue to online parallel presentation. As the information that has been shared before, kindly join each link prepared by the committee and kindly join the link for each room. And please be prepared to all moderator in each room. The first group of online presentation will run about 1 hour and 20 minutes. Thank you very much.